welcome to my channel. So today we're going to talk about this cervical brush. So this will be our case study for today. Then the questions are saying identify this object, and then what is it used for. We have to name the test, then name the site where the specimen is going to be taken from, and then what is the most common virus associated with the cervical cancer. They name the most common subtypes associated with cervical cancer. Then the last question is saying, what are the two, I mentioned two risk factors for cervical cancer. Okay, so now let's start with the answers. So identifying the object. So let's identify the object here. Okay, so identifying the object, this is a rolling brush or a cervical cancer, cervical brush. So that's CB, cervical brush. So it's used it's used as a cervical swab. So as a cervical swab for a pap smear. So it's used as a cervical swab for pap smear. Okay, so it's used as a cervical swab for pap smear. So yes, pap smear. So it's used as a cervical swab for pap smear. Okay, they name the specimen to name the site. So the site where this specimen is taken from, we have this is usually taken from the transformation zone, which is the endo and the exo cervix. So the transformation zone, the TZ, that's where the site is taken from. And then what's the most common virus associated with cervical cancer? So it's HPV, human papilloma virus, the HPV. They name the most common subtypes associated with cervical cancer. So the most common subtypes for that associated with cervical cancer are actually 16 and 18. These are the most common subtypes associated with cervical cancer. And of course, there are others. So others, maybe the one which we don't put here, we have 31 or 45. They can also cause HPV, but so the question asking for the most common subtypes, so 16 and 18 are just fine. Okay, so we have answered this question. Then two risk factors. So two risk factors can have family history of cervical cancer and smoking. Those are some of the risk factors. Then maybe other risk factors we can put to be multiple sexual partners, early onset of sexual debut, immunodeficiencies such as in HIV, chemotherapy, vitamin A deficiency. Then the other risk factors, we can also have uncircumcised partner as a risk factor. Then, so since we talked of the HPV, I also thought of adding more information on this case study. So this case study was supposed to be just on the cervical brush or the rolling brush, but I thought okay, it's important as I also give how exactly the HPV and the human papilloma virus, this one, causes cervical cancer. So how does it cause the cervical cancer? So the HPV DNA integrates into the host genome. Then when it's integrated into the host genome, it starts producing its own proteins. Specifically, the proteins E6 and E7 are produced from the resultant DNA. So what are the functions of E6 and E7? So E6 binds and degrades protein 53, and protein 53 is an important tumor suppressor protein, which means that it regulates cell division by keeping cells from growing and dividing or proliferating too fast or in an uncontrollable way. So that's the normal function of the P53. But this protein E6, it binds and degrades this P53. So when it's binds on, but it's when it's binding and degrading the p53, the cells keeps replicating and the mutation rate of the cells increases. So meaning there is no mechanism to stop them from replicating. Okay, so we talked over. We have finished E6. So E6. So okay, what about E7? What does it do? So what does E7 do? So E7, what it does is that it binds on retinoblastoma. It binds and degrades the retinoblastoma gene. So what's the normal function of the retinoblastoma gene? So it's typically a prototype tumor suppressor gene that encodes a nuclear protein. So we call it retinoblastoma protein that acts as the cell cycle control checkpoint at the G1 phase. So what does this mean? So it means that the retinoblastoma protein prevents excessive cell growth by inhibiting cell cycle progression until a cell is ready to divide, and also prevents cells from responding to growth stimulators whilst inside the G1 phase, the cell growth phase. Okay, so remember we said this same E7, this protein E7 binds and degrades the retinoblastoma gene. So when the retinoblastoma gene is degraded, this excessive growth cell inhibitory factor is going to be lost. So therefore, well, without these two mechanisms we have talked about to slow down cell growth and prevent mutation, that's the one which brings about the CIN, then the malignant transformation occurs. That's the pathophysiology on how HPV specifically causes cancer. So that's what I wanted to add on this case study. And I thank you for watching. And if you haven't subscribed, please subscribe and hit the bell notification. And these case studies, initially we were supposed to be doing them um, as a combined tutorial, but I thought of just dividing the case study so that to make it easy to identify while uh, a specific case study alone per tutorial. So check out other case studies as well, and thank you for watching, and see you on the next tutorial.